Mice are very important in ALS drug development. Preclinical work in the mouse will ensure that future human clinical trials will be conducted better. The mouse can give us a lot of indication about the pharmacodynamic and pharmacokinetic drug parameters. So when time comes to give it to humans, we know how much to dose and how frequently. It will also tell us whether a drug is toxic or not, or whether we should avoid it or not in humans. And finally, it can give us hints about efficacy in an ALS mouse model.
The mouse models that we use at ALS TDI are mostly models that aim to take advantage of findings in genetics in human ALS patients. So the one that we use the most is a model that overexpresses a mutant gene for superoxide dismutase, SOD1. It's the model that we use for most of our drug screening. We have a lot of experience with that model. We've been working with it for about 13 years, and we have a really strong sense of all of the ins and outs on that one. There are other models that we're working on right now in different ways. There are some mouse models that are in development where we're literally inventing the mouse right now. There are other models that have been invented, but now we're actually trying to understand them a little better, where we're trying to understand the attributes that matter. Does it get ALS at all? If it gets ALS, which symptoms does it model best that you would see in the ALS patient? And then we have other models that we're actually doing experiments on that we know enough about to be working on, but not necessarily to be doing full-blown drug screening right now. Most of these models in the development stages that I just described are linked to mutations that cause ALS in genes that are mostly RNA binding proteins. So TDP-43, like the one that we're talking about in this publication here, but also other RNA binding proteins. So there are a lot of advantages to having multiple models of ALS. In this case, one can think about it from having multiple models of mouse models of ALS, or having other types of models of ALS, and that can include new technologies like iPS cells, induced pluripotent stem cells, as an example. But in the case of animal models, what we're trying to do is increase the number of tools in our toolbox for drug discovery and drug development. We know that ALS is a heterogeneous disorder at the patient level. We know that not all patients have the same kind of ALS. They don't all have the same symptoms. They don't all have the same exact disease progression. And we know at the molecular and cellular level that there are differences as well. So having multiple tools gives you the opportunity to see, or to identify therapeutic opportunities that might actually be for broad categories of patients or honed in on specific populations. So really, it's just about accelerating the process of finding drugs for as many patients as quickly as possible.